My name is Gaia Franzetti, and I'm a research fellow at WEISS and Mechanical Engineering Department at UCL. My presentation will be about the interest, application, and manufacturing of phantoms for patient-specific hemodynamic studies. Hemodynamics is by definition the dynamics of blood flow, and specifically it explains the physical laws that govern the blood flow in the vessels. When studying the physics behind it, we consider the vessels as a hydraulic pipe system. According to the location of the vessel we're interested in, we have a certain diameter, inlet flow rate, a certain pulsatile pressure waveform, and vessel wall characteristic, which vary according to the district and the vessel biological composition. We can consider the vessel to be less or more flexible or compliant, which is related to the mechanical properties of the material, as I will explain later. Hemodynamics is widely studied and has proven to be a key in understanding pathophysiology of the cardiovascular system. It has been proven that it plays a role in the development of disease, such as aneurysm or atherosclerosis, and is essential in the design and development of vascular medical devices, such as stents or heart valves. In the past decades, we have seen a shift in biofluid dynamics, like in so many disciplines, towards the so-called precision medicine. Interpatient variability is really important when considering hemodynamics. The anatomy of our vessels can be extremely different, especially when diseased. 3 vessels, like the one here in the image, can have different behavior, impact flow and pressure distributions in very different ways, and therefore affect the outcome of our study. This is why in our group we focus on patient-specific studies, targeting our methods to reproduce personalized features. We do this through our personalized platform. It uses clinical data obtained from a patient to reproduce the precise, correct hemodynamics, using both numerical simulations and experimental models. Specifically, our experimental platform, developed in the past 5 years, enables to reproduce the correct flow and pressure distribution in the vessel replica of the patient under consideration, and therefore our interest in the manufacturing of suitable phantoms. When considering personalized phantoms, you all know that we can segment medical images to obtain a 3-dimensional model of our vessel. When manufacturing a phantom for fluid dynamic experiments, we have to consider several aspects. First, the geometry. We're not considering a cylindrical tube, but we're interested in the intricate geometry of the vessel. And if diseased, it can lead to very complex geometries or difficult challenges. And that's the case of the pathology that we have been studying for many years, aortic dissection, where we have 2 different lumina in the aorta, divided by a thin flap. We then have the vessel material. It can be rigid as a simplification, or try to account to the flexibility of the real vessel. And again, for experimental purposes, it should have specific properties, such as being optically transparent or being compatible with ultrasound measurements. So when manufacturing an aortic dissection phantom for our work, we needed to consider all these aspects, with a specific focus for material transparency, because of the experimental measurements that we wanted to carry out, and the cost efficiency. This is essential in this context, if you consider that when performing personalized studies, it automatically implies that for each case study we need a new, different phantom. So we started considering 3D printing as the first option. Since the vast majority of 3D printing flexible materials are opaque, we considered rigid materials first, as we prioritize transparency and accurate morphology. So we produced the first phantom via stereolithography. This was produced in different sections because of the dimension limitation of the printer. Here in the picture you can see the aortic arch and the section of the descending aorta with the 2 separate lumina. And this is the resulting phantom. We added a connector before the aortic inlet here for compatibility with the experimental loop. In general, the phantom had an accurate geometry. We didn't have problems in the connection between the different parts, but it was not transparent enough for our studies. So we then purchased from Materialize a second phantom with the same geometry, manufacturing with the same technology, in the most transparent material that they have. The phantom was also post-processed to have a better finishing, and the result to be more optically clear, although still not ideal for our experimental measurements. So overall, 3D printing allows us to obtain correct anatomies, but in a rigid material. The manufacturing is fairly straightforward and when produced in-house, cost-efficient. However, it is constrained by the limited choice of the materials, which for us was an issue. As it was mentioned in previous presentations, another very used option is low-score casting. Being dissolvable in water and compatible with 3D printing, we investigated the use of PVA to manufacture vascular phantoms. PVA is usually employed as a support material. With low-score casting, the blood volume of the vessel is first 3D printed in PVA. I will not go into the details in this context, but briefly, the methodology can lead to a box-like phantom with a hollow vessel geometry once the PVA inside is dissolved, or when employing both negative and positive molds in a thin-walled phantom produced with the casting material. As for aorta dissection, we had the added complication of two lumina divided by only a thin wall and connected by a very small hole. We applied the first methodology. PVA printing gave us a really good accuracy in reproducing the complexity of our geometry. Here in the pictures you can see the two different channels and the small connection between them. They were actually printed in two parts to avoid support material problems and then joined. PVA was sprayed with acrylic paint to avoid interaction with the casting material and then placed in a box. An epoxy resin was poured in, and after curing, the PVA was dissolved in water. The resin was chosen as the study was aimed at analyzing low-cost alternatives to known materials such as medical-grade silicone, especially considering the large volume that we were reproducing. Second, we were also aiming at finding a methodology that did not need additional equipment, such as large volume degassing chambers. Having said that, the same methodology was applied by two other research groups at UCL McKenzie, trying different materials, geometries, and dimensions. Briefly, they produced a rigid but transparent, simplified geometry in silicon and a smaller patient-specific aortic arch, but flexible. We all wrote a paper together analyzing the differences and comparing the results, which you can see here below. In general, the methodology is really flexible, but we could not find a suitable combination of materials that would allow us to produce a low-cost, big-volume phantom with optical properties that we wanted. So while 3D printing is limited by the choice of materials, but is able to reproduce very accurate and complex geometries, low-score casting gave us more options, but also introduced additional manufacturing limitations, and sometimes difficulties in dissolving PVA, which might not be compatible with certain geometries. So we thought, what about creating a methodology which combines the advantages of both? Something that would allow us to 3D print flexible phantoms with complex geometries, also controlling the material that we wanted. This would be ideal for our fluid dynamic studies. It would lead to accuracy, realism, compatibility with experimental measurements, and it would be a time and cost-efficient solution. This leads us to the last part of my presentation, which is linked to works explained in the two previous presentations, and in particular to the 3D GelWorks printer. As you saw, it was successfully applied to create ultrasound-compatible organ phantoms. What we were interested to do is to use the same printer to create vascular phantoms, which can reproduce flexible vessel with specific optical and acoustic properties. This is an ongoing project, and as a priority we focus on the mechanical properties of the phantom. In order to calculate the mechanical properties, or stiffness in this context, we needed to consider the so-called Young's modulus E. To obtain this, a tensile test can be performed in bone-shaped thin samples, deformed until the breaking point. We wanted to calculate this in order to compare it with reported values of human vessels, starting with the aortic ones. So we produced samples of GelWorks, both casted and 3D printed, in two different directions, X and Y. And this is because we expected printing-induced alteration of the mechanical properties of our samples, which is evident when looking, for instance, at the printing structures of the samples in this picture. Through these initial tests, we obtained the Young's modulus value from our tensile experiments, and we could verify that the 3D printing and printing direction do affect the properties of the material, as expected. And second, that gel wax is significantly less stiff than human vessel tissue. This led us to the second part of the work, aimed at developing an experimental procedure to design a new, stiffer material, and more in general, to customize the mechanical properties of the material that we want, according to the phantom that we want to produce. First, we chose a set of easily available materials and different weight compositions. Examples are hot glue, silicone, and cornstarch. We then prepared the solution by mixing such materials with gel wax. We gradually added the material to melted gel wax, used the sonicator, performed degassing, and lastly created casted samples. At this stage, we chose to cast the samples rather than printing them because we were more interested in the relative alteration of the properties of the new materials with respect to the gel wax alone, and also to avoid risking damaging the printer. We then performed uniaxial tests on the samples, which allowed us to quantify their stiffness values. At this stage of the framework, the values should be then compared to the target ones. If satisfactory, optical and acoustic properties can be evaluated. The last step of the framework involves the compatibility test of the chosen material with the printer itself. Gel wax therefore represented our reference value, here shown in yellow. In the graph, we can see the Young's modulus value of the material here and its percentage variation of its value here, with respect to gel wax. We then analyzed all the different compositions with respect to that reference. So as you can see, we achieved some control of the mechanical properties with these preliminary tests, both increasing and decreasing the stiffness. However, with these initial results, we're still far from obtaining the desired vessel values. Nonetheless, we further analyzed different samples, and we also observed variability in optical and acoustic properties. Just as an example, the stiffest material, which was a composition of gel wax and hot glue, was also optically and acoustically clear and compatible with the 3D printer. The preliminary results of this ongoing project are although very promising. They demonstrated the validity of the developed framework, of our hypothesis, and the feasibility of customizing the properties of the material. We are currently still testing new compositions and further investigating the possible range of stiffness control that we could achieve. When thinking about the aorta I showed you at the beginning, we can now imagine the possibility of not only creating an anatomically correct phantom, but also a highly customized phantom with the desired specific vessel, or more in general organ, properties, with implication not only for hemodynamic studies, but medical device development or surgical training as well. This can lead in the future to new, finely controlled and highly patient-specific phantoms, relevant not only to cardiovascular work, but potentially to different medical fields as well. And with this, I would like to conclude by thanking all the funders of this work and acknowledging the contributions of all the collaborators in this project. And thank you all for listening.